So the next question is, how do we control for microbes? We can divide the methods of microbial control into two broad categories, the physical category and the chemical category. Fortunately, we are interested in controlling microbes that flourish in our environment because, as you've already learned, there are many microbes that can flourish in very extreme environments. Luckily for us, that's not where we live, so we're going to deal with controlling microbes that flourish here. Heat is the most prominent physical agent, but we can also use radiation, filtration, ultrasonic waves, or even cold. In this section, we're going to look at some of these methods and their applications in medicine, commerce, and the home. We'll first begin by looking at heat as an agent of microbial control. Generally, elevated temperatures are microbicidal, and lower temperatures are microbistatic. We can use either moist heat or dry heat. We'll cover both of those here shortly. Moist heat is generally more effective. Moist heat is provided by hot water, boiling water, or steam, and the temperature ranges are from about 60 degrees Celsius to 135 degrees Celsius. Its mode of action is through coagulation and denaturation of proteins. Dry heat, on the other hand, has really low moisture content. It involves an open flame or an electric or infrared heating coil. They range from 160 degrees C to thousands of degrees C. The mechanism of action for dry heat is dehydration, denaturation, as well as incineration, literally burning things to ashes in very high heat. In this table below, you can see a comparison of times and temperatures to achieve sterilization with moist versus dry heat. When we think about temperatures, we can have moist heat at 121 degrees C for 15 minutes. Should sterilize most anything. In dry heat, that same temperature of 121 degrees C would need to be up for 600 minutes. As we've already established, bacterial endospores are going to exhibit the greatest resistance to microbial controls because destruction of the spores requires temperatures far above boiling. The vegetative states of fungi and bacteria are least resistant to both moist and dry heat. However, they do vary in their heat sensitivity to both dry and moist heat. Viruses are surprisingly resistant to heat. However, heat can be a really effective means for taking care of them. For practical purposes, all non-heat resistant forms of bacteria, yeasts, molds, protozoa, worms, and viruses can be destroyed by exposure to 80 degrees C for 20 minutes. Now the question is, how much time at what heat is necessary? High temperatures generally allow shorter exposure times, and low temperatures generally require longer exposure times. The thermal death time is noted to be the shortest length of time required to kill all test microbes at a specified temperature, whereas the thermal death point is the lowest temperature required to kill all microbes within 10 minutes. So TDT is at a specified temperature, and TDP is in a specified time of 10 minutes. The TDTs have been experimentally determined for microbial species that are common or important contaminants in various heat-treated materials. Many perishable substances are processed with moist heat in this fashion. Many remain on the shelf at room temperature for years and years. The quality, the speed, and the cost of processing needs to be considered. For example, in canning, we're considering the removal of Clostridium botulinum spores. They must be killed, and this can be accomplished by using infinitely high temperatures. However, if we use infinitely high temperatures for an infinitely long amount of time, the food inside the can is going to turn to mush. So we have to figure out what an optimal TDT is to maintain the quality of the food, as well as make sure that the job is done. So there are several common methods of moist heat control. We can use steam under pressure, non-pressurized steam, pasteurization, and boiling water. Let's begin by looking at steam under pressure. In order to do this, we need to understand a little bit about the relationship between temperature and pressure. As you probably already understand, as we If we increase the pressure, we will increase the boiling point of water. At 1 atmosphere of pressure, which is 15 pounds per square inch, water boils at 100 degrees C, and the steam that it creates will stay at that temperature. By increasing the pressure, we increase the temperature at which water boils, and thus we increase the temperature of the steam produced. When the pressure goes to 30 psi, that's 15 psi above normal, then water boils at 121 degrees C, and thus the steam temperature is 121 degrees C. As you're already aware, boiling water at 100 degrees C is not capable of reliably killing all microbial spores. So we use an autoclave. The autoclave is a machine that allows us to generate pressure and boil water at a higher temperature, thus allowing for sterilization. The most effective temperature and pressure combination is 30 psi, which yields 121 degrees C. This is 15 psi above our normal atmospheric pressure. This combination allows us to kill all endospores. And keep in mind, it's the temperature, not the pressure, that kills the microbes. Now, home pressure cookers are built around this same principle. Not only are we allowed to permeate what's in the pressure cooker with high pressure steam, we're also able to sterilize food in a pressure cooker. If you have a pressure cooker at home, that will be a great way to sterilize things in the home. Now, we can also use non-pressurized steam as a means for sterilization in a process called tyndallization. This is intermittent sterilization. In this technique, we put our things in a rack inside a steam chamber and expose free-flowing steam for 30 to 60 minutes. After this, we've probably killed all the vegetative microbes. However, some spores remain, so if we incubate the sample for 23-24 hours, and allow for some of these spores to enter their vegetative state and multiply, we can treat again. We'll do this procedure for 3 days in a row. A single exposure is not sufficient to kill all the spores, but allowing them to incubate for 24 hours may cause many of those spores to enter a vegetative state. Again, we kill them with the heat, steam. Again, we allow them to incubate. This is used for some canned foods and certain media preparations that we'll use in the lab. But because the temperature never gets above 100 degrees centigrade, very resistant spores or highly resistant spores that don't germinate could survive the whole 3-day non-pressurized steam procedure. So it's not as effective as pressurized steam. Another couple of moist heat techniques we have are pasteurization and boiling. Beverages such as milk, fruit juices, beer, and wine are easily contaminated during the collection and processing procedures. Heat can be used to reduce the microbial load and destroy pathogens within these beverages. In pasteurization, heat is applied to the liquids to kill potential agents of infection and spoilage while retaining the liquid's flavor and food value. Pasteurization incorporates special heat exchangers. There is a flash method and a batch method. The flash method exposes the liquid to 71.6 degrees C for only 15 seconds, and the batch method exposes it to 63 degrees C or 66 degrees C for 30 minutes. The flash method seems to be preferred because it's less likely to change the flavor or the nutrient content of the substance. Both means are effective at inactivating most viruses and destroying the vegetative states of most bacteria and fungi. However, not the endospores. Interesting to think, though, even after all of the pasteurization happens, milk can still contain more than 20,000 microbes per milliliter. This is why unopened milk can still spoil. Now, sterile milk is available. It can be produced under ultra-high temperatures. It only takes 1 to 2 seconds of exposure and allows for a shelf life of months. But this is really dead milk. And some say that living dairy products are much more valuable to our system than the pasteurized or sterile milk. The primary targets of the pasteurization method are things like Salmonella and Listeria and some of the Mycobacterium species, as well as a couple of enteric viruses. Another technique is boiling. However, this is just for disinfection. It's certainly not sterilization, but it can be used in the home and clinics very effectively. The idea is to expose materials to boiling water for 30 minutes. This will kill most non-spore forming pathogens, including resistant species like staphylococci. It's also a technique that we use for unsafe drinking water because it does kill most of the microbes and protozoans that could be found in it. Now, let's move on to the dry heat mechanisms. First of all, hot air and incineration. Incineration ignites and reduces microbes to ashes and gas. And as you've been practicing at home, it's common in the microbiology lab to use incineration on inoculating loops and needles using a Bunsen burner or candle in our case. The flame of a Bunsen burner reaches 1870 degrees C at its hottest point. Now, the candle, probably not quite that high, but still pretty effective. More recently, we've seen an increase in the use of tabletop infrared incinerators to take care of sterilization in lab environments. Hospitals and such use very large incinerators to burn all sorts of materials, including bedding. Now, a dry oven can also be used. Usually it's an electric oven. It has coils, much like your oven at home, that radiate heat within an enclosed compartment. Exposure of 150 degrees C to 180 degrees C for 2 to 4 hours is effective for sterilizing heat-resistant items that don't sterilize well with moist heat, just based on their composition. Dry oven sterilization is good for things like glassware and metallic instruments, as well as powders and oils or waxy substances that the steam cannot penetrate well. Before we move on to look at cold and desiccation, let's have a quick concept check. What temperatures does an autoclave achieve at 15 pounds per square inch, or 1 atmosphere above sea level? Now remember, sea level is 1 atmosphere, so really the total PSI is 30 PSI. But because we're already at 15 PSI, then you crank up the autoclave another 15 PSI, the total PSI would be 30. And we just covered this, so it's a pretty quick one. 121 degrees C when we set the autoclave to 15 PSI.